Hi, my name is Sierra Waite, and I'll be presenting over question 25. So first I want to briefly go over all of the different techniques, and I've organized them into this table. On the left, we have electrophysiological techniques, which mean that they measure the electric field that's generated by neurons. And then on the right, we have the two metabolic techniques, which mean that they measure the metabolic costs as neurons consume oxygen. So we'll start on the left with single unit recording, and this measures the level of the individual neuron. It uses a microelectrode to detect voltage spikes generated by action potentials, and so it is an invasive process. And as you can see on the bottom for each of these, you can see if it's a high or a low temporal or spatial resolution. Next, we have magnetoencephalography, or MEG, and this measures the magnetic fields produced by electrical currents in the brain. It is a non-invasive process, but it is not commonly used in research, but it is used in clinical settings. Next, we have electroencephalography, or EEG, and this measures the electric voltage across the entire scalp, and it is a non-invasive process. Then we have positron emission tomography, or PET, which measures blood flow using radioactive tracers, so it is minimally invasive. And finally, we have functional magnetic resonance imaging, or fMRI, and it also measures blood flow, but it is a non-invasive process. So first we're going to go over EEG in a little more detail, and it measures the electric current through electrodes across the entire scalp. So whenever there's activity to certain areas of the brain, synaptic currents are going to flow into the cells in this area. This flow is not symmetric though, so it creates a dipole, which is just the net current flowing in one direction. And you can see the dipoles in the image on the right. They're represented by the arrows. And the dipoles parallel to the skull are best measured because, as you can see, the dipoles in the folds of the brain are pointing to each other, which means they cancel each other out. So the EEG can analyze these dipoles and determine the location of activity, which is source localization. And the activity is represented by waves, and I've attached a picture of some of the waves at the bottom there. Next, we have PET, which measures changes in blood flow. So a radioactive tracer is injected, and then the scanner detects the gamma rays that are created from the positron emission from each of the voxels of tissue. The tracers will collect in areas that have more blood flow, so this will show higher activity by lighting up on the PET. Finally, we have fMRI, and it indirectly measures blood flow by detecting oxygen levels. So it uses a bold response, which is just the ratio of oxygenated to deoxygenated blood. So the ratio increases whenever there is increased blood flow, and more active areas require more oxygen, so more blood flow is going to increase. So blood flow is going to increase to this area. And the bold response is correlated with the average energy consumption per voxel. So now I'm going to go over some of the challenges that are presented with these techniques. First, we have to know that these techniques show correlation, but not causation. So each of these techniques collects observable data where something happens and then we observe a response. But we have to know that that response is just correlated to the thing that happened, and we cannot assume that it was caused by the thing that happened or vice versa. Then we have some indirect measures. With PET and fMRI, we are directly observing some sort of response or signal, but then we have to connect it to an indirect measurement. So for example, we can observe a bold signal, but then we have to connect it to brain activity in that area. We also have the issue of distinguishing excitation and inhibition. We can't really do that with these techniques because they only show activity changes in the brain, but they don't show what kind of activity change, so we don't know whether areas are being excited or inhibited. Next, we have some issues with noise. So one of the problems is partial volume, and this is where one voxel can cover multiple areas. So the smaller a voxel is, the weaker the signal strength is going to be. So sometimes to get a better signal, the voxel size will be increased, which means it covers more brain areas, but then this means it might pick up on multiple different areas or multiple different cells. We also have the issue of co-registration, and this is because these techniques are used on different people, so we need some sort of way of averaging it over multiple people. So we lay their images on top of each other and kind of distort them to align them so we can compare different landmarks. Finally, these techniques only measure functional connectivity. So they only measure the correlations between the activity levels in different areas of the brain. But we're more interested in the effective connectivity, and in order to do that, we have to solve the inverse problem, which is a lot more work. And that's all I've got. Thank you.